I'm Juan Manuel Lopez Smith. I work as research associate and NMR facility manager at Pontificia Universidad Católica del Perú. We are the only NMR facility in the whole country. We have a very nice 500 megahertz with a cryoprobe, but we are very interested in to develop methodology that can be translated to clinical facility. NMR is a great analytical tool. It gives quantitative measurements, but also structure measurement. It's very accurate, robust, reproducible. So the idea is to develop some tests that can be used in clinics. For example, we are developing the drug susceptibility assays that can be used in clinics. We have recently developed an assay to determine resistance in Mycobacterium tuberculosis by NMR. Tuberculosis is still one of the top killer diseases worldwide. At least 1.2 million people die every year. The treatments are long with side effects. The emergence of multi-drug resistance and extensively drug-resistant strains really complicate the treatment of the patients. And we are very interested in a drug that's called pyrazinamide. Pyrazinamide has two particularities. One is molecular mechanism still unknown, but it's used for more than 17 years. There is a debate of exactly what is the mechanism, so it's very difficult to develop other drugs for the target because we are not sure which one is the target. That's one particularity of the drug. The other particularity of the drug is that there is no reliable drug susceptibility assay that can be used in clinics. And this is due to a second peculiarity of this drug, is that a neutral pH, commonly the Mycobacterium assay are based on bacterial growth, but the neutral pH, the drug is inactive. We need to decrease the pH so the drug can become active. And then the pH inhibits the bacterial growth. So that's why there is no easy drug susceptibility assay. Something that we know for this drug is that indeed pyrazinamide is a prodrug. It enters the Mycobacterium tuberculosis and it's hydrolyzed to its active form, pyrazinoic acid. So all the resistance mutation found in the bacteria are in PNCA protein. PNCA is an enzyme that catalyzes the reaction. So one way to assay the susceptibility is just by measure the ability of the bacteria to produce or not pour pyrazinoic acid. So by NMR, we can very easily follow the transformation of pyrazinamide to pyrazinoic acid. So by just measure the concentrations on the supernatant, we can just follow the kinetics. So the idea now is, can we link those numbers, the rates, to the susceptibility of the strain? So for that, we have collect a sputum sample from patients. Indeed, I have an extensive collaboration with University Cayetano Heredia in Peru, and they collect regular samples of sputum sample. So now we use NMR to just follow the transformation of pyrazinamide to pyrazinoic acid, and then we can link this rate directly to the susceptibility of the bacteria. So we can know if the bacteria is resistant or not. Bacteria susceptibility means that the drug is active against the bacteria. So if the bacteria have resistance, the drug will be not active. However, in the case of pyrazinamide, what we saw is that the mutations indeed confer a resistance by decreasing his ability to form pyrazinoic acid, so the active compound. So the drug enters the Mycobacterium, but it's not hydrolyzed to the active compound. And that's what we measure by NMR. In recent years, the development of benchtop equipment that has a high resolution, now they achieve a very stable magnet. I think the line width is around 0.4 or 0.2 hertz. This is a good opportunity to translate some experiments to something that can be more automatic, high throughput and can be easily done by non-expert, and also the equipment is quite expensive. So this, the benchtop equipments are really an opportunity to try to make NMR more accessible to everybody and translate it also to clinics. Metabolomics is a hot research now. Indeed, the ability of screening the metabolome and trying to find some correlation with disease or infection or whatever, it's really nice. So NMR is very reproducible, robust, and needs not manipulation of the sample. So we can repeat very easily the experiment. However, one of the limitations is the sensitivity. Nowadays, if we have application with molecules that we know and the sensitivity is enough, it's translated that to a benchtop equipment will make it more accessible to everybody. However, I think in future, the idea is to improve the sensitivities. And I think in future, benchtop equipment combined with methods that enhance the sensitivity, so we can get similar results what we can get with a regular spectrum. I'm really interested in develop methods that can then be translated to clinics. So I think NMR is robust and we don't need to handle too much the samples. So that is great to translate it to clinics. So we need to make methods that are simple, and also how to analyze the data. So if we can make it automatic analysis of data, and now with artificial intelligence that can be done, so now we can translate it very easy to clinics. Clinical application, most of the time, we are quantify specific metabolites. But indeed, the whole spectrum has information. So, but the problem is you need to disentangle what are the variabilities related to some disease with just the variabilities between patients, or just the regular variability in humans. So it's important to disentangle that. So artificial intelligence can do it. If we train it, they can do it for us. So we don't need to spend time to analyze the data. We can just use artificial intelligence for that.